In this video, we will discuss about Angelman syndrome, and this disease is uh, related to genetic mutation, and the children's or patients are generally have the genetic mutation by birth. Symptoms of this Angelman syndrome is that the children is unable to move or find difficulty in balancing uh, it find difficulty in balancing themselves. Children finds difficulty in balancing themselves who have this Angelman syndrome, and they are they also find difficulty in crawling. So what happens in this case is that children receive one chromosome from mother and one chromosome from parents, from not parents, from father. So this mat represents the maternal or chromosome from mother, and pat represents the chromosome from father or paternal. Chro- So what happens in mat is that the PWSIC is methylated, and because of the methylation of PWSIC, the DNA methylation at this site, it will affect the transcription of its neighboring gene, and those are mentioned in the gray color. However, the transcription of this UBE3A occurs without any problem, but it caused the problem in the antisense transcription of UB3A, this methylation. And what happens in case of paternal chromosome is that this region is not completely methylated. However, the region is methylated at different sections around this area, and because of this, the neighboring genes are able to uh, transcribe as it is shown in the blue color. However, the antisense strand of this UB3A is not been able to transcribe because of the CPG island presents in nearby this area of the paternal chromosome. So we can see that thing in the shade in the this shaded arrow or shaded white arrow or faded white arrow. So the transcription of UB3A is not completely, it's not completed also in this case, in the case of paternal chromosome. And this is the zoom in video of this is the zoom in uh, view of this paternal chromosome is that the truncated 5 prime UB3A is formed. However, the antisense strand is completely silenced because of the CPG island available in nearby sites, and the 5 prime is also in some case the 5 prime is also not been able to transcribe if it is in if this person, if UB3A gene is not properly functional or it is mutated. However, in normal case, the 5 prime strands of UB3A on the other strand, that is on antisense strand, will be able to on the sense strand, yeah, will be able to code itself for the antisense strand. Then it can be transcribed with the help of RNA polymerase 2 into the mRNA. So, however, in case of in case of Angelman syndrome, what happens is that there is a mutation in the UB3A gene, or this complete deletion of the UB3A gene that leads to this problem, that is Angelman syndrome. And the third model shows why this why this UB3A transcription problem arises. There are two different kind of model it shows. First one is the collision model, that is RNA polymerase 2 one RNA polymerase II is transcribing the one side of DNA, and this RNA polymerase II is another RNA polymerase II is transcribing another side of DNA, and because of the collision of both of them, the some part of this UBE3A antisense strands unable to transcribe, whereas one side or one side of the DNA, one strand of the DNA will be able to transcribe properly. And another model is that RNA DNA interaction models. So what happens in this case is that when the RNA polymerase is transcribing one strand, however the other strands is not being able to transcribe because of the modification of the DNA, or histone modification of the DNA. Because of this, the transcription uh, abort at intermediate location within the DNA. So this RNA formed from this strand will not be functional. Uh, so this is how the UB3A transcription and translation occurs, and this mutation on this gene will cause the problems that might lead to uh, uh, problems in balancing themselves, or they might even find problems to move properly. The children might find problem to move properly. So I hope you have understood something about this Angelman syndrome. If you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.